Is it a glass sponge, a Rosalida?
No, this one that we're looking at is a demosponge.
Okay.
We think it's Halicondrididae family. We don't know which species it is, but we have subsampled it.
I'll make a note that the first one is Halicondridae.
Mm-hmm.
Started to apply the dye.
I'm not sure. Patty, can you tell us when?
Okay.
Or if you're on the other side of the sponge, maybe we'll see it better. I don't know if you can come to the opposite side. So Patty is the one controlling the arm. This is a delicate procedure, so it takes a bit of time.
Jigsaw. Den andra då? Ja, så mycket.
Ay, a squid!
Oh, yeah.
Oh, hello!
Oh, there's a lot of current.
Wow! Stay with us. Little squid show.
Yeah.
Beautiful. If it comes closer, we can even identify it. Okay. Come back.
Thank you for dropping by.
Yeah. See you later. They're interesting animals in the water column. Chiropods and squids.
Yeah, the summit is extremely rich.
When we had landed, there were several grenadiers on top of this. Here in this area. Look at all the anemones and soft corals, encrusting sponges. There's so much going on.
And they're all living on previous dead sponges. So when the sponges die, their skeleton of glass remains at the bottom and little by little creates a very thick spicule mat. The spicules are the names for the skeleton of sponges. So they accumulate at the bottom, and all of this biodiversity grows on this spicule mat. So on top of the dead sponges.
It's really pretty. Also some amphipods rushing through. I also really love the fan-shaped sponge. How it's...
Yeah, this is probably Cladocroce.
Okay.
We collected it in one of our first dives, so we didn't know what it was.
But was now, it the one that was quite spongy when we collected a piece?
It is. It is. At first, I think we thought it was a glass sponge, but it's a demo sponge.
Okay. So glass sponges have glass spicules.
Yep.
And demo sponges do not.
No, they do as well.
Aha.
So there's 4 groups of sponges: demo sponges, glass sponges, calcareous sponges, and then a little group that was described in 2012 called homoscleromorpha.
Yeah.
They are not very present here in the deep sea. So it's essentially demo sponges and glass sponges that we see.
Okay.
The calcareous sponges are also present, but they are minute and very well hidden. You
We found a couple, didn't we?
have found a couple on rocks or actually in the mud that you've been sorting.
Yeah.
You've been finding them sometimes.
Yeah.
So they're there, but they're just very small and cryptic.
I see. So what we're currently waiting for or trying to do is to get the dye through the hose out in the water, and that hasn't succeeded yet, but the pilots are working on it, and this is like just how it is with all scientific experience. I'd say that you plan something.
It's typical of a scientific.
Very typical. You plan some, and especially in the deep sea. I mean,
Yeah, it's difficult to take into account the pressure, maybe the temperature. It can really play. It might seem insignificant, but to get the liquid out of hose, it can be important. So.
Right. Because the pressure of the high pressure we have here at this depth, it squeezes.
Yeah.
And it might block the hose somewhere.
Here
where
the
it wasn't
pressure
blocked,
is 60 times the pressure at the surface. So it is high pressure. We would not survive on the top of this mountain if we could not dive there. It's impossible. So that's why we send robots. They can cope with the pressures, with the very cold water. Let's see.
0.0.
0.0. Well, 0 degrees, pretty much.
So it seems that the first trial of our experiment is not working, unfortunately. There's nothing we can do about it.
No.
Basically, it's just we have to accept for what it is. That something is blocked, and we can't do anything about it now, unless it sort of loosens. But I don't know. It might be that we have to give it another try another day.
Yeah.
Which is a bit unfortunate, of course, but again, there's nothing to. That's how it is.
Yeah.
That's just how it goes. We have so many pretty animals to look at that this would basically have been a nice add-on to this dive, but maybe we can fix it.
Can you look up on the HD just up to the loop on the top of the hose?
Top of the hose.
Yeah. Is it green just with the back? So it's coming up, is it? Zoom in. Is it getting there? Yeah,
To zoom on the tip.
yeah,
Or
just
yeah.
zoom in. Just want to see the back of the hose here. Oh no, turn to the right now. Keep on right. Is the green going up?
I don't see green.
No. But so the hose is yellow at the end?
Yeah, but the hose is.
No, like itself, the material is yellow at the end, right?
Yeah.
Okay.
Keep going. Stay straight on. Keep going.
It's not getting up. So is it gone? Look up the top. Go back to your right top. To the right. Not there.
Yeah.
Go down.
So we're having lots of different cameras here in the shack, and what the pilots are doing are they're checking the other cameras that you currently can't see, and they are searching along the hose to check whether the liquid that we want to deploy or expel, where it has stopped in the hose or what's going on. So that's what's happening. It's actually quite cool to see the angle of all the different cameras and all the equipment that you can actually see with them that is mounted to the ROV.
It's black back there.
Maybe we still managed to fix it.
So I think Patty's going to try something new.
We're going to attempt to squeeze the bag.
Okay.
So I think we are going to use the other arm. These are highly technical movements that we're doing in the deep sea.
Yeah, we basically have a bag of this dye in.
Oh, it's coming out!
Ah!
Did you press on the hose or
Yeah.
on the...
No, sit back.
Ah, on the
Bag.
bag.
Okay, so the
Okay.
hose is not blocked.
No.
Oh, look at it.
So yeah, I should remind you, fluorescein is actually a natural compound. We use it a lot in medicine actually.
You have to use your end, you want to use the G4.
If for some diseases with the bloodstream or diseases in the brain, when we want to actually monitor if a compound is crossing the blood-brain barrier. So we use these types of compounds in people's bodies. Here we're using it in nature to see if we can track the water coming in the sponges.
Let me get into position.
I hope I was too quick with my judgment. I really do. I really hope I was too quick. We can do
Yes.
that.
Would you rather be that it's a sponge farther away so that you can...
But
no?
is it here, Paco?
If you can get a little bit closer here.
I mean, by the lasers.
Yes, exactly. And if we could turn off actually the lasers, that would be even better. Thank you, Rob.
In like that?
Yeah, a little bit outside of the sponge.
Okay.
If you can, yeah,
Like there?
yeah, here should be good.
Okay.
Oh wow, it's happening.
One more across. Just shoulder forward. Then watch it. The strike comes up at the same time. Shoulder forward.
again. We're going.
With this packer, we'll move that out of the way in a minute.
Yeah.
It looks
I'm just
funny.
explaining that to the viewers that we're using the other arm to push on the pouch that is filled with dye to stimulate the release of the dye. So this is why you see the two arms in action right now.
Oh, I'm really like.
I hope you're all crossing your fingers or holding your thumbs. In Sweden, people hold their thumbs.
Oh, look what I do. How do you say in Swedish?
Hålla tummarna.
Hålla tumlarna. Okay.
Yeah, hit straight down.
I'm crossing and holding and everything. Oh,
It's
It's
yes.
coming out. Yes.
Yeah, off you go.
Yeah. So let's see if some of it is taken up by the sponge.
Okay, I'll take away my my arm out of your view, Paco, and we can come back if you need to.
Sorry?
I'll take my arm away,
Okay.
and we can come back in if you need to.
It moves towards us. If we place it here,
Yeah.
it moves through the sponge. Look at the Assyrian.
Yeah, you can see an Assyrian that has taken it up in the bottom of the image. But I don't think the sponge has taken it. So what you have to know is that sponges are animals that are alive. So we've been close to this one for a while. It could have and could have decided to stop pumping, which is maybe why we're not seeing any results. When you handle sponges, when you move them, and so on, they decide to stop feeding usually. Maybe this one sensed with currents or because we touched it also. When we touch a sponge, it tends to stop feeding. So here, yeah, I don't think it worked. Should we try in another place?
Another location or
Yeah.
a sponge? I can reach further out if you need to.
Oh yes, let's see, because there was the white ones.
Mm.
Can you reach these?
What is it? Back? Yeah, let me see.
We're going to try to reach these large glass sponges in the background.
Point to me where I should go on our screen.
Yeah, exactly. Here or here. Or maybe if you are in the middle of the two,
Yeah, if you want to.
that could be
Do
a
your
solution. I would say this looks very much like a scientific experiment. The first try usually doesn't work.
Yeah, yeah. But I'm very happy that we're on it. Like we have the dye.
Hmm.
It can be pushed out. That's really good.
Try to get it as close to the base of the punch, but without touching it.
Yeah,
Yeah.
okay.
So viewers should know that this hasn't been done in the deep sea so many times. This is not an easy experiment to do.
You're going to be full reach back now.
4 now.
This looks
Okay, 1 second. I was looking at the thing there.
So the pilots are adjusting
No, hold on.
adjusting the arm to position both the arm at the correct spot
Yeah.
at the bottom of the sponge, but also untwisted so the hose can run free, or like the dye can run free in the hose.
Ooh,
Oh, that's a nice decapod.
purple one. In shallow waters, it's much easier because you just go with a syringe filled with fluorescein, and while you're scuba diving, you just really sit at the base of the sponge. But here, it's much, much more challenging.
A little bit scooch forward and we'll have it then.
Okay. So we're gonna move the ROV a little bit closer so that Patty has a bit more space and flexibility to reach those two glass sponges. This one would be really nice.
Yeah. I'm really hoping it works. I want to see it. I really want to see it.
So for those joining us now, we are on the sponge ground at the summit of Schulz Bank Seamount in the Arctic, close to the mid-Atlantic ridge. We're in this beautiful, beautiful sponge ground, very colorful in the deep sea.
So I was just laughing because the things start flying in the water column. It will settle again, but it looked a bit funny.
So now we're a bit closer. So let's see. We're gonna wait. We're gonna set the syringe in place. Wait for the dust to clear out.
It's interesting because it doesn't look that sedimented, but
Yeah,
is.
but there's probably lots of little fluffy.
Zoom in for me there, Paco, just so we see where we are.
Do you want me to zoom?
Yeah, please.
Yeah. So what you also have to know is that a lot of sponges love to bud. So when in the event that we break a little piece of sponge, I mean, some sponges really like that. Like this, they can have a clone of themselves, settle somewhere else.
Is that close enough, Paco, or more?
That seems close enough, absolutely.
You're happy?
Yeah, yeah.
Okay.
now we're going to push the bag again. Now,
So now with our second arm with the hose, we're going to push down on the pouch that contains the dye. And then of course the arms will come back to their places so that you can see with us if it works or not.
mm-hmm.
We can see all the details of the grip of the left arm.
how mechanical.
Yeah, it's pretty amazing actually.
Yeah,
Just behind there.
it is.
That's coming, keep going.
Yeah, I can see it now.
There it
I can
Yeah.
see it.
Okay, take it off now, yeah. I think we're too far back.
Yeah, well, it seems to be a bit too far
We
back.
can work on the front ones because the current is coming this way.
Maybe zoom
Yeah.
out a little bit, Papa.
Sorry?
Zoom out a little bit.
Yeah.
Guys. Then the sponge takes it.
Yeah,
Tilt the
it really
picture.
depends a little bit on the current.
And then zoom in.
It's your circle,
I mean, the Obsidian has taken it up,
Yeah,
that's for sure.
yeah,
And then there's some lurking here.
yeah. Not sure where this is going. Then maybe. Yeah, maybe we need to be a bit
Yeah.
more on that side because it seems the current just pushes it.
Okay, so we'll try that.
I remember us planning this. You suggesting like that we should do this, and in my head,
Of this glass sponge. These are all the incoming water openings. So they're like thousands of little mouths taking in food. And the large opening on top is the excurrent water opening called the oscule.
was that the one that you had under?
That's the
Yeah.
same one we collected
Yeah.
a couple days ago.
Yeah.
I think it's Schaudinia rosea, one of the really common glass sponges here.
Do you have it in the list?
Yes, I think I... no, I just put Roselidae.
Okay. How do I write it? Schaudinia?
Schau.
Oh. Okay. Schaudinia and then
Rosea.
Rosea.
Yeah, exactly.
Okay. I really love this sponge. So pretty.
Mhm. The glass sponges live really in the deep sea, so to be able to do an experiment on the glass sponge, I'm not even sure it's ever been done before. In the deep sea. We want to.
Yeah. And it's funny because it doesn't change a thing if
No,
you
but we
tilt
still-
your head, but
Roll the arm over your genitals so you can see underneath them. Go clockwise and forward. It's coming alright. What about it? It's just lighter. Yeah, no, it's lighter.
uh,
You're in the image. So, there. Zoom in, Paco.
Yeah.
oh,
This should be a good location.
I like how it wiggles a bit.
Mhm. Could we try to put more dye?
Yeah, we can, yeah.
Yeah.
I think the location was quite good.
The location
Because
I think is good now.
it didn't-
Yes. And also, I mean, it could be distributed to
Right,
any of the other sponges.
right.
But maybe we haven't put enough, so the dye dilutes very rapidly because, of course, there's so much water.
Yeah, right.
But I think the color of the dye is not an issue because we see it when it comes out.
Yeah, very well.
It's a good dilution, but because of the current, maybe. Oh, there we have a nice view.
More?
Yeah, more.
Wow.
Fixed up.
Yeah.
Load the picture.
So we've pushed for a bit more. Ooh.
Wow.
I think you can see it on the front, right?
Yeah.
Yeah.
This one, yes,
Yes,
probably.
so that's
Yes, you can see it on the
the one closest towards us.
yeah, the glass sponge that is on the right.
Do you want to zoom in?
Yeah, I can zoom in.
Hey, do you want me to move out of the way, Paco, as well?
No, but the left ones are
no.
stop.
Yeah.
The left ones are starting as well. Just you see both of them.
Yes. Yeah, you can see movements coming
Wow.
out, especially the one in the center. I mean,
Big ones.
this one. You can see it's coming out of it now. So it's been through it and now it's coming out of it. And you can see it for these three here. It's very visible now.
Oh, this is so pretty.
Look at this.
Wow. Oh, it's working.
Oh, look at this.
Oh. Oh, wow. I need to take
They're alive.
Yay.
They're alive.
And you can show it.
Yeah. Very nice.
Wow.
They're up.
Wow.
Just a small video.
We have to thank Paddy and Rob because
Aw.
this was team's work.
Three of us. Andrea came in.
Oh, Andrea.
Wow.
Thank you.
So you can see the current is not super strong. It just flows quite slowly.
This is so cool.
If you turn off the lights, do you see it?
It's not
Yeah.
fluorescent, the
Is it
dye.
fluorescent? I don't think so.
Um,
To turn on the light, would it be?
No, you have to have UV light.
Okay.
With UV light.
But we can do the stage light. Yeah.
So will I take my hand away now? 'Cause it looks like there's more still coming.
Yeah, I see that. Yes.
Yeah, I'll take some more away.
Yeah.
Okay. They're on you for me.
This is so cool. You can really see it coming out of the osculum.
Yeah.
Wow.
Lads, tell me when it stops. I think it's stopping.
I've never seen anything like this before.
Looks like we're on Mars.
Yeah. Beautiful.
So they're still pumping.
Free flow now of the
Yeah.
Again, I'm gonna zoom on
See if I can stop it.
the main.
You can really see the dye
Yeah, here.
around all the openings.
Yeah.
Shadow. That's it. Stop.
So these animals are constantly filtering the water, cleaning the water, eating all the bacteria, or tiny, tiny organic matter and feeding themselves with. And then they release the water that is pretty much cleaned of everything they have taken. We think they also release compounds this way. We're not sure what these compounds are used for. Maybe for communication between sponges. That would be so cool. I'm quite sure one day we'll find such compounds that enable sponges to have a common behavior at some point, maybe release their reproductive cells or warn of a predator and so on. There's a couple of research groups in the world looking into this. So this is what we call the field of chemical ecology, trying to understand relationships between species through the light of chemistry. Wow, this is
I'm enjoying this. It's so special to see it. Wow. Really cool. I'll take the 100th picture now. I mean, we will
it's like a little
have stunning pictures from
chimney.
our HD afterwards, but
Let's see if we can zoom in a little bit more.
wow. It looks almost unreal because it's so clearly that they suck it in and then expel it again, like really actively.
Yeah, and it's also a good way to show that there's a lot of currents down there, because we see as soon as it leaves the sponge, phew, it's taken up.
Yeah.
And I mean, apart from the movement of the animals, it's difficult for us to realize how much current there is. With smoke or a dye, it's perfect.
Yeah. Oh, I'm very relieved that this, in the end, that this worked and that we are seeing this.
I'm hoping Julio and Sergi are watching this in their bed.
These are our two colleagues on board.
Yes. And in our cabins, we all have a flat TV, and if we put on channel 4, we have exactly what the ROV sees. So I know that when we wake up, one of the first things we do is turn on the channel to see which dive, what's going on.
Which is it's so unlikely. Like it's I would never do that at home.
No.
Wake up and turn on the TV, and here's really the first thing I do. Just as you said, to watch what's going on. Wow.
Well, it's not too bad.
Still shows.
Okay.
Can you show the lower one as well?
Sure.
I like the one lying down. So that's
It almost looks like it has a little leg standing.
Yeah, this is really what I wanted to show you today. But otherwise, we're going to explore.
Yes.
We're going to continue to explore. We're going to be a little bit limited in sampling because, well, you saw, the other arm is well equipped for another purpose today. But we still have a slurp.
We do. We do.
And the slurp is really, really useful for lots of organisms.
Oh.
Yep.
Oh, the little one is still pumping. Oh, and yeah, it's not over yet. Wow. It seems also really like that it sticks around. Like.
Is the arm blocked? There's no more coming out, or?
I'm up above you. Just so it doesn't contaminate.
Oh.
Okay.
Oh.
We have enough for another experiment if you want it.
Yes, that would be great. Another spot. Yeah, absolutely, absolutely. Yes, we can lift off maybe and explore a little bit this magnificent sponge ground. I mean, this is a unique habitat. I think we haven't seen this much sponge density and soft corals and anemones and everything. Ascidians
It is
covering everything.
Schulz Bank is so special,
Yeah.
so rich. I mean, honestly, this should be protected. This should be.
Yeah.
We need to save these environments. They are. I mean, they are so rich, and they're fueling the other seamounts in the area.
Yes, and creating super rich habitat for many other creatures. I mean, that you don't see on the screen, of course. A lot of baby fish, a lot of adult fish that come to feed as well. So these are nurseries for a lot of different organisms, but also
Yeah.
all the vertebrates. Yeah.
Sorry. Taco, have you an area close by you want to go to, or should it move it quite a distance?
Well, we can move quite a distance, maybe.
Okay.
I don't know. A couple of. I would like to find one geodia, but I don't see any. So if we can move a little bit more, maybe we can spot one on the way.
This was successful. Wow.
So we're lifting off from the bottom now.
For the first time, which is a bit unusual after 1 hour. So it's a very special dive today. It's really, really special.
Just don't come back great. The arm sticking up the front.
Look at the size of the sponge ground.
Incredible.
So there's a lot of scientific literature about the seamount, which was explored for the first time in 2008 by a Norwegian ship, the Geosars, during a Norwegian expedition. This was the same expedition when Loki's castle was discovered. So I was on that ship as a PhD student. We explored and discovered Loki's castle, and then we moved to Schulz Bank. And these were the first images of the sponge ground and the first collections at that site. So this is, my second time back.
There's another squid.
Well, where?
There.
Oh, yeah.
There's another squid. Do we still see it?
Is that
Yes.
it?
Yes.
I don't know if it's a bit too far.
Yeah, a bit too far away. Not only rich in sponges, also rich in squid. Very nice.
For instance, I see really large ones here that could also be good candidates. Yeah.
What's that? It's another squid.
Mm-hmm.
Oi, it's the third one.
Maybe this one. In the distance would be.
Wow. Okay, that's the highest frequency of squids we've seen so far. Wow.
I mean, this
And
sponge
there's
ground
another one.
is truly,
Look,
it's truly rich.
this is squid number 4.
So it's this one in front of this one. If you can,
Wow.
we could do another experiment.
Incredible.
Uh-huh.
Is this Asconema foliatum?
Very good. Very good.
We have been at sea for 3 weeks.
I know. You know all the names. It's really good. I don't know all the names of the amphipods. Far from it.
Well,
So-
you are a good teacher.
Okay.
Okay, so we are in facing Asconema foliatum. This is also a glass sponge. It has a more intricate shape, but let's see how it behaves with fluorescein.
Maybe we want to tilt like a tiny bit. Yeah, amazing.
Zoom back up.
Yeah,
Yeah. Happy there?
yes, maybe a little bit more behind, according to the current.
Mm-hmm.
This is also a glass sponge. You can see it's very thin. It's very fragile. And
Maybe a bit lower.
yeah, could you go more at the base of it?
Mm.
Yeah. So let's see. Usually it takes a couple of seconds, maybe a minute, to start seeing it reappear.
That position okay back up?
That position should be good.
See more of it, is it? Give you more flow if we need to.
Mm.
Maybe we need to inject a little bit more. I don't know if we can. Do you now have control over the quantity or not really?
A little bit, yeah. We don't have to use the second arm.
Yeah, yeah. Let's try that.
Okay, so we'll come back to
Are these two different species
Yeah, I'm not sure.
or they're
But
younger ones?
yeah, I feel sometimes the soft corals they differ a bit in their habitats and the way they look. But it's hard to tell. This potato is also interesting to see.
I was looking a little bit
Yeah,
outside
yeah.
if other
Yeah.
animals picked it up.
Mm.
The advantage of glass sponges is that they are pretty large and they're white.
They're starting, don't they? Or does it just looks like
I
it?
think it's more coming from.
Yeah, I gave it another little bit there.
Yeah.
Oh, okay.
We need to go further around the back, or is that okay?
You mean more
No.
dye or?
No, I can give more dye, but I think I'd be too close.
Yeah.
Do we need to go around the top?
We can try. Yes, let's try that.
It's interesting how some of the water just
Yeah.
sinks,
Yeah.
stays at the bottom.
Yeah. Absolutely. So again, I remind our viewers that this is a dye that is used in medicine on humans. It's not dangerous, and it's called fluorocein. And we always use it when we study the filter feeding activity of sponges. That's the first thing we do when we have a sponge in an aquarium and we want to see if it's alive. We give it a bit of dye to see if it's pumping. If it's pumping, it's alive, and it's actually pretty happy. So it's a way to test the happiness of sponges.
Really? So the previous one was quite happy,
Yes,
and this one is maybe just a bit sleepy.
it's possible. Again, we actually don't really know when sponges pump or not. It seems they may have some cyclic activity. Sometimes when they pump a lot, sometimes they maybe sleep or take a rest. So maybe that's the case with that sponge.
Or
It's difficult to know.
The cilia just beat slower, or maybe yeah.
It could be as well, but then I'm expecting we would see things
Yeah,
coming out.
Yeah. Yeah, right. But it is funny how it just sort of aggregates.
That looks like it's gone all around the base there now, doesn't it?
Yeah.
It's in that species that we found all the ovipods. You remember?
And lots, I do. How well it looks from the inside.
It looks very beautiful. But it's not pumping that much.
No. No.
Would you mind to zoom in couple just for the interest of the
Yeah.
looking for one?
Give it another push with the other minute back over it.
Yeah.
Yeah.
Let's give it one more try on that species.
This is also nice.
So
I agree.
maybe the image will be blocked for a while by the second arm, but that's okay. The second arm is used to push on the pouch that has the dye and enabling to push the dye out.
Someone calling the RV shack.
Yeah, Paddy, here at the bridge, can you hear me?
Go ahead, yeah.
Yeah, Paddy, I want to change my heading there to wrong spoke, which is the 30, with that swing there.
2:30. Yeah.
Is that June? Yeah.
Yeah, that's a geode, yeah. But they're so covered with
Yeah.
with stuff, but actually they're pretty much everywhere.
We can't really see the oscula though.
No. That's why I think the glass sponges are probably the best.
We had, was it yesterday? I think so. When Sergio was here, we also had really pretty Julias.
White, very white.
Not very white,
No?
but white, more white than these. Like
Yeah.
a bit less sedimented or covered with speckle mats.
So it was maybe stellata, because there's also stellata.
Okay. It had one,
Like on the-
one osculum.
No, then it was not stellata.
But I remember very well the moment when you showed us both Parva and Tancelli next to each other. That was really nice.
They reached us so 200 in it.
Yeah, you just give us another couple of minutes, Baz, before you do the second bit of that.
No water, standing by.
Cheers.
This device is so cool for the pilots to handle the manipulators. It's really impressive.
So they have in front of them like a mini arm
Yeah.
that they twist, and which directly controls the real arm of the robot.
And you have different movements depending on if you want to open or close the jaw and
Yeah.
really.
Yeah, it's fairly complex.
Yeah.
Yeah, I see it coming up.
Oh wow.
So we're injecting higher quantities to see if
Zoom in right in, Paco,
Yeah.
and move back if I have to. Just get down a little, do I? Am I okay?
Yeah. There.
there.
It started.
Yes. There.
Nice.
You can see it on the side here. It's coming out. I mean,
There too.
Yeah, there too. So
Can you do some backs and start to see where
Sure.
more and more companies come?
I think this one has also started now.
I mean, maybe every little piece of sponge has its own circuit in a way, so that's why we're not seeing it
Yeah.
all over the place. Maybe they don't communicate, but we saw it a little bit.
Yeah. Some of them were pumping.
Yeah,
Maybe you're right, and they take turns.
yeah. I mean, we know absolutely nothing on especially in glass sponges which live in the deep sea that you can't cultivate in an aquarium.
Yeah.
We know basically nothing about the
Want to give it another
way
press?
they pump. No, I think we're good.
Okay.
I don't think we can get better than that. We saw it a little bit,
Okay.
so it was nice.
Yes.
And
It's
then
also nice to see how the water stays in some
Yeah.
areas. because
as it's been like trapped in the spicule map
Exactly.
in some way.
Exactly. And it gives you some idea on like how larvae would maybe also be.
That's it.
Would you mind zooming in on the signal to the right?
These ones?
No, the ones on the
There?
Yes?
Yeah.
Just
Yeah.
to see, because I think it also took up some of the
Yeah,
line.
you're right.
Oh yes. Oh yes. Yes. Yes.
Look at these ascidians.
Wow. This
So
looks funny.
they are also filter feeder, and I think what we're seeing is the branchial sac, if I remember correctly, and there's a brighter. There's two brighter places there, and that's the endostyle. It's the place where the ascidian produces mucus to trap food and bring it down to the stomach. Which makes sense then to see it highlighted with the dye, because that's where food accumulates at some point.
Yeah, that's good.
Beautiful.
Thank you.
Yeah. Well, it's nice to see also on other.
Oh look, it's still there.
Yeah.
The like the dye in front of this mini cave-ish structure.
Mm.
It takes a long time to disappear. Wow. I find this highly fascinating because I have been thinking lots and lots about how larvae distribute and how it can be that some species are so widely distributed while others are not, and how the water masses, how it's all working, because obviously they are transported with water masses, but how well are they actually transported and where do they get trapped, sort of thing.
Yeah.
That's really interesting.
Are we ready to move on?
Absolutely.
Yeah.
When you're ready and happy,
We're ready to move on.
Okay.
and we're on the fly.
Yeah. I'm gonna zoom out a bit so that
This is just incredibly beautiful. Could we put the lasers back on, please?
Yeah.
Thanks.
so the lasers are 10 centimeters apart.
We're chuckling because we have been saying in previous live streams that they are 20 centimeters apart. That was sort of fake news. And we are very happy to correct this now. Sorry for the confusion, in case you have been watching several live streams and suddenly it's 10 and not 20 anymore. Wow, that sponge is really pretty.
Yeah. Yeah. So the large brown ones, we
Okay,
think,
guys, whenever you're ready there.
is Halicondridae sponge. We don't have a name for it yet. We're still working on that. And all the white ones belong to the same family, Roselidae, same family of glass sponges. And the very intricate one here that
Wow.
we've been doing an experiment on is Asconema foliatum. The little orange balls that are a little bit everywhere is also a sponge, and that's Craniella infrequens, the little golf ball sponge. And then you see a lot of massive brownish spherical sponges. They're sometimes a bit buried in the spical mat, and these are geodias.
I basically just logged everything you said. Lots of sea stars, different species.
A lot of soft corals as well, and ascidians.
Yeah.
So the big potato brown ascidians you see sitting on the ground.
And-
They're also very abundant here.
Yeah, they're both, sometimes they're brownish, but also often translucent almost. So you have to develop sort of an eye to see them well. Here's some lollipop
A
sponges.
lollipop sponge,
Stylocordyla.
Stylocardia borealis. Absolutely, you can see the oscule, the opening, main opening quite well.
Maybe not increase the water.
I think that's right.
Oh wow!
Fine, thank you.
I love the big sea stars.
What about those sponges up there, Paco?
Yeah, we could try to reproduce this. Yeah, this looks like a little sponge city, sponge suburb.
Right, come in from this side.
We're going to try to reproduce our dye experiment with
Not really, just the side on there.
these Rossellidae sponges.
Otherwise, they need to be up under this one. It takes a lot of them
Wow,
through
the
hoses.
big one.
This very big.
Ooh.
Yeah.
But here in front, this is also quite big already.
Yeah.
And we're doing a little dance here now.
We're trying to place ourselves with respect to the current because we want the dye to go at the base of the sponges. We don't want the dye to go directly in the current. And there's also a lot of yellow sponges. All those yellow patches are encrusting sponges. It's Hexadella detritifera. That's the scientific name. Every living organism that has been described on our planet has a scientific name made of two names: the genus and what we call the species epithet. So for us humans, it's Homo, that's the genus, sapiens, that's the species name. And it's the same for every living organism now on this planet. And this is thanks to Linnaeus. Linnaeus is the Swedish scientist in the during the 18th century that decided to name everything on our planet. And people since have been continuing his work. And we are some of these people.
Yes.
We're describing new species, giving them scientific names in order to document biodiversity on our planet.
Do you mind centering it a bit?
No,
Thanks.
not at all.
You can turn the lasers off when you have a hand. Thank you.
Oh, they're on. They are off.
I say just turn them off.
Turn
No,
them
they're off now.
Yeah.
All good.
So this seems to be a Rossellidae, probably Chaudinia rosea as well, even though I would need to check the skeleton to be completely sure, but it has an extremely beautiful shape. It's So if you're joining us now, we're on a little experimental trip right now, trying to see sponges feed. Probably you've never heard that in your life, this idea of seeing sponges feed. Because they are animals and they feed to survive. They need some food and they get the food from the water. And so they're constantly filtering this water. This is one of the ecosystem services that sponges provide for free, cleaning the oceans.
Shall we go a bit more down?
That position okay, Paco?
Are you right on the ground?
No, I'm just above them.
You're just above it.
Come up higher if you wish.
No, at the base is perfect.
You zoom in there, you have a look in your camera.
Yeah.
Tell me.
Oh, this looks good. This looks good. Yeah.
You might need to give it
The
Right.
current is tricky because it's actually not going
Yeah,
this way.
we might need a bit more dye actually to
Yeah,
Next we get some more dye.
yeah.
And
look at the fish.
Let's look at the fish meanwhile.
Okay.
Can you see the fish on the left?
It really dilutes quickly, huh?
Yeah,
Cool.
yeah. And I mean, when we prepared the solution, Julia and I, it was pretty dense and concentrated.
Okay.
We even thought it was too concentrated, but
Oh,
actually, when it's in the water, it just phew.
See the way those purple bits, they're going to the left and the dye is just there. You know, they're wafting to the left.
yeah.
Yeah, so hopefully when the dye comes, it should get around the boat.
Yeah, yeah. That's why I think it's a matter of quantity, so that it's
Yeah.
a piece of it at least goes to the sponges.
Mm.
Yeah, it's catching. Yeah.
Yeah, this is very challenging to get enough dye in the water column. But I mean, the previous one worked super well. You can really see it's
Yeah.
very nicely. I love how much there's going on here on Schulz Bank.
Yes.
So diverse,
Yes.
so rich.
It's like a tropical reef.
Yeah, it's incredible.
I mean,
It's catching on the front. The top. Your arms out. Shoulder down. Yeah.
which means there's a lot of food, because
Right,
to feed all these animals, you need a lot of food.
yeah.
Shoulder down. Shoulder down.
Speaking of which, I think our dinner was really lovely today.
Shoulder down.
It was,
Yeah.
and my appetite was back.
Yes.
That's good.
That's good.
I've been fighting a little bit with the sea conditions yesterday, but no, I'm back.
Back in business.
Back
Oh,
in the
that's
lab.
good.
Yeah.
I think this is a different species from this.
Yes.
Yeah.
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 Look at the baby lollipop.
Oh yeah.
It's even smaller than
Yeah.
the golf ball
And
sponge.
Canela is already
Yeah.
like,
Yeah. So we are currently in the command center of the robot, which is in a container on the deck of the boat. So we're moving with the boat, actually. We can feel it now and again. And the robot is connected to the boat by a very strong cable.
yes.
And that's why robot and boat must move together in order to not lose the robot, not get entangled in the cable. So there's constant communication between the bridge on the boat and us here in the robot command center. So for instance, because while we're doing experiments, we told the boat to stop waiting.
I like how you call it Robot Command Center, aka the shack.
The shack. This is how we actually call it between us. But it is, I mean, you don't see, I don't know how many screens there are. 12,
1, 2,
13, 14, 15, 16 screens.
16 screens. on how many square meters? What are we talking about? Like 10, 12? I don't know, 10?
So
Yeah.
it's not the NASA command center, but almost. Almost.
Yeah.
Okay, so we're getting
Oh, there we go.
More.
There we go. Let's see.
When you go closer with the other one, am I okay there?
I think you're okay because now it's going in the right direction.
Yeah. Say when you're happy.
Yeah. As soon as we see them.
Oh, this looks good now.
Yeah.
Promising. Promising.
Yeah.
Yeah. I think that one has started, but I'm not quite sure.
Yeah. Okay, let's stop there
Right.
and see.
Ooh, that's a lot now.
Yeah.
That should really work.
So this is fluorescein, a dye commonly used in ecology or even in medicine for humans. So it's a way to see the flow of liquid. So that's why in humans we use it for bloodstream, to see, for instance, if blood, if some compounds get inside the brain with the blood or not.
This has started. It's just
Yeah.
hard to see now because.
But these ones are definitely
yeah,
red and green inside. So let's see.
Yeah. It sort of sticks around.
Yeah, I mean, but the two one in the foreground, I would say are pumping, but not as much as the previous glass sponge we tried. So maybe this is a slow pumper, or it's just resting. Yeah. No, I don't think we will get better than that.
Yeah, the first one was.
But the first one really worked very well.
Yeah.
What about the ones there?
Let's see what's in the back.
The big one.
Which ones can you reach?
No, the two in the back, are they bringing anything through?
Oh,
Ah,
With my eyes.
you mean these ones?
Yeah.
No, I haven't noticed anything.
no.
There.
Like, it's true that when you do this experiment in the aquarium, I mean, there's not that much current, so everything stays. Here, it's taken up very rapidly, so you have to be really quick.
Can we zoom out a bit?
Sure.
Just thinking to. Yeah, no, but then we have to resettle and maybe we move on and
Yeah.
if we see.
Let's move on, and maybe we'll see another
Right.
sponge that could do the work.
Yeah.
Okay.
Thank you, Patty. And so there's actually no rocks on the summit.
Grenadier.
Okay.
My dear, it's a grenadier.
Should have died that.
Hmm?
Should have died that.
Yeah.
Chair.
So you've got one panel for the right and one for the left arm? Ah.
The kids are behind you, right?
Can we turn the lasers back on, please?
Sure.
Thank you.
They're on.
Thank you.
That's the mode.
Grenadier.
It's pretty
There's another grenadier.
slightly over your side.
Oh, what did you say?
Yeah. I
Ah.
think it's the same one that
Yeah.
was there
Yeah.
earlier.
Okay.
So you know you
So the
you bring it back from 0.
coffee.
Coffee.
Yeah.
Do you want the
No,
Ooh,
Oh,
actinophore.
very nice.
Yeah. So we're reorienting ourselves with respect to the boat and with respect to our transect, because we always go in the water with a plan and a direction that Jenny and Julio have been working on really hard. Actually, it was quite the steep learning curve.
Oh yes, for both of us. Yeah. And lots of
this is RV.
Can you send out this Excel in case we find something interesting?
Yes.
Yes. So you were
yeah,
saying.
no worries. Basically that there every day you have to discuss with everyone. Okay, this is the transect that we have planned. What do you think about the profile? Do you think it's doable? Do we want to do it? Do we expect hard bottom? Do we expect soft bottom? Do we think that this dive will represent the faunal diversity that we maybe expect, or that we want to explore, or that we went to seamounts that were completely unknown to science, that where we believe no one has ever been before, and it was quite challenging to plan the dives and to find cliffs that represent the biodiversity that is actually found down there, because even though we dive for many hours, it's just a needle in the haystack what we cover, really. When you look at it at the map, when you zoom out, the distance seems so little, but when you're here, you see how much we actually observe on this couple of meters, because the diversity is so high that we get a good idea of how this ecosystem is structured and what kind of species live down here.
So we're back on the Schulz Bank sponge ground. Very rich in sponges, but also soft corals, anemones, ascidians, sea stars as well. They're a bit here and there,
Definitely.
but they're definitely
Different species of sea stars. We have different species of fish, quite high density of grenadiers.
and we are 640 meters below sea level.
Yeah.
This is not the deepest we've been to. This is the top of the mountain, but it's probably one of the richest places we've been to.
In case you haven't watched our live streams from the previous dives that we did on Schulz Bank, I highly recommend that you do. It was one of the highlight dives, I'd say, with this beautiful diversity of all kinds of benthic animals. Really, really impressive to see. So this is again the Halicon.
Yeah, Halicondridae.
Yeah.
Yeah. It's a shame we don't have a name for it because it's super common.
Yeah.
It's but again, I think for this one we'll probably need the help of genetics. So all of the specimens we collect will, at least the sponges, will come to the Museum of Evolution at Uppsala University, where we will sequence, we will read some of their genetic code and DNA to help us identify the group to which they belong.
Oh,
to.
what's that? Also sponge, but like
Yeah, that's a weird shape, but yeah, it's a sponge.
it's very interesting.
Yeah.
So there's one sea star species here, and then to the right, now out of our picture, but I saw it earlier. Yeah, there we go.
Oh, there.
It's two different species of
Yeah.
sea star. The right one, I believe, is a tilasta, and the center one that is now moving out of the picture is another species of which I currently do not know what it is.
And all of these species, all of these communities are living on spicule mats. Spicules are the glass pieces that make up the skeleton of sponges.
Can we have a vessel movement of bearing 030 at 0.1, please?
Asconema. It's really beautiful.
This is fast, slow.
No, this is good speed.
Thank you.
Yeah, thank you.
This is perfect to observe and.
That's good.
Mm-hmm.
Oh, this is. Shall we sit and try it? Or do you think it's too?
Are you ready to try another time with the dye?
If you
Yeah?
want. Yeah.
Should we try this one?
Yes.
It's rather big, it's a bit isolated from
Yeah.
other sponges.
It looks like a saxophone. You know, the shape of it.
This one. Yeah.
Yeah.
Which are we?
It's again the
Chaudinia rosea.
Sorry, we have to stop the movement for now.
As I said earlier, there's no stones on the sponge ground, so everything that looks like a stone that you can see on this image here, there's a lot, are actually sponges as well. They're just covered with, we call it hair, it's not hair, it's actually very long spicules. Again, the glass needles that make up sponges. So they're covered with those spicules that retain sediment, that's why they become brown. And then on top of that you have other sponges or other organisms that live on them. And that's why they seem to be colored with yellow or white. They become the substrate for other organisms. And then you have little transparent little pouches here and there, also on this image. These are ascidians. They are actually quite close to humans. If you look at their evolution.
Yeah, they're also chordates,
Yes,
just
exactly.
like we are.
Yeah.
So ascidians are much closer to fish than all other animals that we see in this picture. Although it doesn't really look like it. They just look like a translucent little pouch.
Could you remove the lasers?
They are chordates.
Thank you. So we have also taken, we have also put out our sextant camera. It's like a fisheye camera with a 180 degrees wide view. In order to get a different shot of things, a different perspective. You can't see it, but we are filming it, and this footage will also be used.
Oh, there we go.
I think we. No? Yeah, you might need to put more.
Is that okay here?
Yeah, the position is pretty much perfect, yes. So Andrea has positioned the syringe just behind one of the two tubes. And now they're going to try to push on the pouch containing the dye in order to have a bit more dye coming out in one go.
It would be nice if this one's pumping. Do you think it's one that...
I think so, because if you look at the roots, they have common roots. So I think this is one individual. Yes, definitely.
One individual with two oscula.
Exactly.
Okay. So it's pores where the water comes in and osculum where the water expels.
Exactly.
Yeah. I have the feeling that every time we take a photo with our telephone here in the shack, it's like a tourist picture because you know what I mean? It's like we have much better footage, but still we need to take
our own pictures.
our own because it looks so nice.
So here's more. Let's try to see now if it's picked up by the sponge. So Rob is moving the other arm out of the way.
Oh, it disappeared so quickly.
Yeah. Let's see. Let's wait a couple of seconds.
The position is really good of the.
The position is super
syringe,
good.
So.
Yeah. Yes? Yes?
You can see it?
Or is it behind or? It's difficult to know if it's coming from behind or...
Yeah. I agree. We were really lucky with the first one then, I have to say. Wow.
But we really did put a lot.
Yeah.
Much more maybe. Maybe that's the key as well. Could we, I think the position is perfect. Could we try to push some more dye out of it? Okay, we're gonna try one more time. No, let me zoom out a little bit.
What's that?
Sea star. Maybe there's a little drama going on there.
Little drama. Yeah, when they first start feeding, it's more like feasting than feeding. These sea stars. They just extend their whole stomach outside of their body and just go quite excessive. Oh, we have poked an Ascidian. Okay.
stay with your pen on that.
Yeah. Because there needs enough to go to be taken by the sponge to really see. So let's just stay there for a moment. I think it's okay.
Yeah.
Yeah.
Can we take one more? No, that's good.
Okay.
Yeah,
I think this one is
Yeah.
pumping.
we could even take it away.
Could you take away the
Yes.
syringe so that we can see better?
I think you're right,
Yeah.
but it's hard to tell.
Yeah. It is hard to tell. Let's see.
Since it all moved in that direction, if it's not pumping, we can move it.
But here I think it's coming out from the large one, no? I think
Do
this
you want to
is,
zoom in a
Yeah.
bit more? I think this one is pumping now. No.
I think. It's tricky.
Yeah,
It's tricky to be
Yeah.
100% sure here.
it was more clear with the other one.
Yeah.
We all agree that that was definitely active pumping.
Mm,
Here, there is some, it expels,
Yeah.
the top one expels like a bit. Not that much, but there is something going on.
But it's also it's difficult to know if it's coming this way.
Let's check the top one again, maybe.
Mhm.
That one was. It just sticks around again.
Yeah, I guess a lot has. Sorry.
No worries.
Yeah.
Yeah. Look at the obsidians. I like this patch to the left, the green patch on the bottom.
Hmm.
It's nice.
Or maybe actually a lot of individuals are not pumping during certain times. It's difficult to know, but this is interesting. It's a first try, so someone should try to replicate what we just did, because that's how science works, right? You need to replicate, ideally by someone completely independent, with a different technique, if they can see the same result.
Whenever you want to lift off,
Yeah.
I am.
Yeah, I think we can go.
Yeah. Just want to get rid of the controls.
Well, was worth another try.
Yeah.
Somewhere else.
Nice specimen.
If we haven't got any left in the bag, then obviously we can't.
Mm.
There should be plenty left. So it's not a successful one.
Hard to see.
Yeah.
How about that one?
A big one like that?
Or
Yeah, I mean,
you might have a better eye than me for.
maybe this one.
Okay.
So it's coming up on yours.
Yeah, sure. I'll move over close.
Or the geodias that are just in the center.
Yeah, but I
Mm.
might be a more tricky on the geodias.
Mm.
Usually we do it on sponges that have a barrel shape with
Yeah.
a really large opening, because then it's much more visible.
Maybe these are not so good candidates then because they are flat, rather flat.
This one you mean?
Yeah.
No, not necessarily.
Okay.
Because there's pores on all sides.
Yeah. Ooh,
Ooh,
the rock is moving.
or it's probably on the geodia.
Oh yeah, sorry. It looks like a rock, but
That's a
Yeah,
steep.
yeah, it's a
Yeah.
bit steep. Okay,
Okay.
let's move on. It's difficult for us to know how steep it is. The pilots, they see it because of course they're driving against the current and the topography, but for us it's not always easy to
to
see
estimate.
this.
Yeah.
Yeah.
Can we resume past movements?
Wow. So nice to
Thank
see.
you.
Could we fly a bit closer to the bottom? Would that
Yes.
be possible?
Yeah.
And with the lasers on.
Thank you.
Thank you.
See, if you have a look at our cliff cam here,
Yeah.
coming up quite steep.
Oh yeah, okay.
What that means is the centre of the sub is literally between 1 and 1.5 metres off the seabed, but the front of the vehicle is only centimetres. So it's a little bit tricky to try and navigate up.
So you're flying like this?
No, the other way around. We're flying like that, and the hill's coming like that. So the front of the vehicle is
It's
there,
closer to
and
the sea.
the cliff is
Yeah.
coming down.
Thank you for trying to fly close,
Yeah,
even though
but if I
we're
fly too
facing.
close,
No, no,
I land up ploughing.
no! Don't plow.
So that's why I say it's a balancing act.
Ooh, lots of Anemonen for the autumn.
Yeah. Lollipop sponges.
Yeah.
Lots of Assyrians.
Dense spectromat. We have a tape change in 5.
Okay, so
It's a pretty anatomy. We're a bit silent because we're scouting for maybe another sponge to sit down, or basically just observing the fauna.
Oh, lovely.
It's interesting because now the density of soft corals is a bit less.
Yeah, and it's more asconemas really everywhere.
And to the top left, top right, we get more
Soft
soft
coral.
corals again. Yeah.
Yeah.
So it really depends. They are sensitive to lots of environmental factors that we can't really see and that we don't really understand yet either. It's not only like depth is always a factor that influences faunal assemblages and species, but there's so much more. Inclines, currents, temperature maybe, but however, no, temperature is quite stable. But current activity, anything that impacts the larval settlement and food availability. Oi, that sea star had just had a big dinner.
Big feast.
Very big dinner. Look at it. Tiny arms, large belly.
Grenadier.
Oh yes. Thank you for flying so slowly. This is really nice.
Yeah.
Good to see and scout for more
We're
sponges.
going up.
Yes, we are.
Yeah, going up now.
We're at 634 meters at the moment.
Like a lateral to port, and there's more.
It's quite steep to sit here. But what?
But the density,
Wow.
That's fine.
that's crazy. Again, imagine that everything is sitting on a spicule mat and most of the glober-shaped things you see below, brown, are actually living sponges or ascidians. So it is completely packed. I mean, I guess they must compete for space,
Oh yeah,
some of these filter feeders.
yeah, I believe so too. Can we do a tape change, Andrea?
Yes. One, two, three, stop.
Thank you.
On the bottom left is a 1990s.
One. You ready, Jim?
I'm ready.
1, 2, 3, recording.
Thank you. Could you let the dry lab know when you have time? Thanks.
Try live RV.
My lab, we heard you changing tapes.
Thank you.
So there were the first 2 hours, huh? It was like
Already?
boom. Yeah.
Wow.
I mean, with a bit of settling in the beginning, but basically, yeah.
So earlier we were told that the average viewer of these videos stays on for 8 minutes,
Oh.
which is actually pretty good.
I think so too. I think so too.
Because that means some people will stay definitely longer, and people will stay maybe 1 minute.
Yeah.
So an average of 8 minutes or so is decent. Hoping that they will have learned something from the deep sea, from sponges, from the
Yeah.
Arctic.
And people might tune in again at another time, because these videos stay on YouTube and
Yeah.
can be rewatched, and
Absolutely. Yeah.
they will be there. I mean, we all know how it is with like day-to-day life when you're not on a vessel. You have so many other things to do and going on that you can't sit hours and hours in front of a live stream. Usually. So, thank you for everyone that was watching.
And thank you for your comments.
Yes.
There were really a lot of interesting comments, challenging us a little bit on on the identification of things. But it's also a good way to realize that these habitats are poorly known in general. We can say it. Still lots of species undescribed to be discovered, and just understanding basic biology about these Scythian sponges, anemones and such. We're missing a lot of just basic knowledge about their reproduction, their distribution, the symbiosis that they have with bacteria, many things. And I can already tell you that later on we will have 2 scientific researchers who will comment in Spanish. So you should come back if you want to listen to them in a couple of hours. We are several nationalities on board, all European, I would say.
Yeah.
But it's a mix of languages on board. A lot of German, a lot
Right.
of Spanish,
So French.
some little French, a lot of English, of course,
Norwegian.
Norwegian,
Swedish.
Swedish.
Ooh.
Yeah,
Portuguese.
Was that a sapphonivore?
maybe.
I'll... Portuguese. What else?
Irish, English.
Irish. Scottish.
Yeah. Welsh.
Welsh. Right. I think that's it.
Mhm.
Maybe. Wow. Can we just hold here just for a sec?
Of course.
We don't have to sit, just hold, because
Oops.
I want to do one nice zoom on these anemones.
No worries.
Quite high density. Thank you. It's always so nice to the eye.
Bridge ROV, can we stop for a moment, please?
So some of them have their tentacles out to collect anything that might drift by in the water column, and some of them don't.
What is that? Sorry?
I assume it's just like with sponges, that it's not always that they are active and that they need to feed, and they're saving their energy. They're taking a little break. Everyone needs a break from time to time.
Next to the anemones, you have a lot of little brown balls. These are Geodia henscheli. These are very common species in the Arctic.
Do you mind tilting up just a bit?
No?
Thank you. Can we zoom more or are we maxed?
We can zoom more.
Yeah, I just really love that one. It's so pretty with the pink tentacles. Yes.
We have a tiny polymastia
Oh.
with the two papillae. This is Polymastia thielei.
Yeah.
I'm
and-
happy with this composition.
Yeah, me too. We have a good balance here of anemones
Yeah.
and sponges. Lasers off, please. Can have a little beauty shot here.
Thank you.
Thanks.
Yeah.
Oh, I'm also
yeah.
quite happy. I like it. And this is a very representative image of Schulz Bank,
Yeah.
where we have the soft corals, the ascidians, the glass sponges, the anemones. A sea star would be nice in addition, but apart from that,
A Dumbo octopus,
yeah, come on,
but it would not fit. So
no, squid. Oh, but Schulz Bank is just very, very pretty. And don't forget, we are in the deep sea, and
yeah, the deep sea is not empty.
no,
It's full of life.
all right. Thank you for stopping.
Thank you.
We can continue with the lasers on, please. Thank you.
Pretty Jeremy. Can we have vessel movement 050 at 0.1, please?
That was nice. It's a little dense field there.
Thank you.
Where was your first cruise ever?
I have to think back quite some years now. My first cruise ever was definitely a cruise with the University of Bergen, or whilst I was studying there. Yeah, it was the Mariano cruise. Yeah, I remember. I did the bachelor in biology at UIB, and in the third year we had a complete year, like 60 ECTs that we could just choose whatever we wanted to do with them, which is amazing. So in the first semester I chose 2 courses that both had 15 ETC, and one of them was a practical, and I was very free in what I wanted to do. It had just I needed to get it approved, but then I could basically do whatever, and I went to I approached the Mariano project. That is a very large mapping project for biodiversity in the Norwegian Sea. And asked them whether I could maybe join them and maybe even join an expedition, because I really wanted to explore this sort of side of the biology and go to sea. And they said yes and took me on the GeoSARS vessel, and we did the mapping of the biodiversity on the seafloor, and it was the very first time that I I got in touch with this hands-on taxonomy to identify species whilst you look at them on a camera. And yeah, I learned so much during that cruise. I still use this knowledge today. So
Cool.
yeah, that was really a very nice experience. Yeah.
How long was it? Was it a week, 2 weeks?
It was maybe 3 weeks. It
Oh,
was
wow.
not a short
Oh,
cruise.
wow.
No,
Yeah,
it was quite
that's,
long. 2 or 3 weeks, something like that. I don't really remember. It was in 2017. That was a nice one. We saw lots of snow crabs in the Barents Sea. I remember that. These large crabs.
Yeah.
And lots of other things too. How about you?
That was probably my cruise with a German ship, Polarstern.
Oh.
And we went to northern Norway to study deep sea coral reefs. And I was in charge, of course, of the sponges.
Wow.
I was a PhD student at the time. And I've used a lot of this material for my PhD. But I'm still studying the rest, especially with my postdoc Julio. He's studying it extensively, compiling everything and trying to identify as much as possible. And this was collected in 2007.
Wow.
That was
Okay,
a long time ago.
another 10 years back, yeah.
But it's a rich collection. Still, that will bring a lot of results. And it's a really nice comparative material because it was from the coast of Norway, the deep sea. And it's really good comparative material with what we're getting now in the Arctic, because there's always been this idea that the Arctic fauna is different from the continental shelf fauna of Norway, for instance. But there's very few studies that have really tried to link sister species in both environments. So that's something I'm very eager to start working on now that I have this material from the Arctic to compare with.
In case you wonder what all the red little dots are that are swimming in the water column, these are amphipods, little crustaceans, and there are tons of them. It appears that, you know, they're just out and about swimming.
Probably good food for fish, or
maybe even some small squid that hunt them, and we find them everywhere. Like, they sit on the sponges. If we zoom in and focus on the small things, we see them everywhere. Sometimes some jellyfish, but mostly amphipods that drift by, swim by. They're pretty good swimmers. Many of them are very good swimmers.
so many Asconema. Wow. Asconema are the cauliflower sponges you see a little bit here and there. They're very abundant here.
I'll log a couple. And Craniella.
Yeah, a lot of golf ball sponges. The round orange ones.
How about the dye experiment? Do you want to give it another try?
We could, but I think we need to be somewhere a little bit flatter. So I'm still waiting for that hill to flatten a bit.
Yeah. The folder is upstairs.
With the profile.
Yeah, I can bring it later.
We don't have it here now.
Maybe not. You know what? I can swap and then get it, because I'm also interested in the profile of the dive.
Okay.
So I will be.
I forgot it. Shall I run up again and get it?
You can.
The white folder.
Yeah,
I
we
was
wanted
supposed to
to look at the profile of the dive. Thank you, Anna.
Shall I take the tape up while I'm at
Yes.
Yes.
The routines are really set now, huh?
It's been a steep learning curve as well.
Oh, definitely. Yeah. Do you mind if I tilt it down a
No,
bit?
please do.
Oh, that's a nice sea star.
Mm-hmm.
It's again another one. Look, that one is again another one. I think 0.1 degree is almost the warmest water temperature we've had so far.
Wow. Yes.
That's still super cold, but
So here we're in a different water mass than the water mass at the bottom of the mountain. So you have to imagine that the sea is not uniform, it's not homogeneous, it is several layers of different waters that are not mixing, they're on top of each other. And so if you're an animal living in water mass A, it can be very difficult for you to disseminate your larvae in water mass B. There's a real border there.
Can we increase the speed to 0.2?
And so during this expedition, we're also collecting specimens to do population genetics of sponges and amphipods to try to see where these borders are and if they are still permeable to some genetic connection or not.
yeah. Oh, thank you. I can. Shall we swap or?
Up to you. Yeah.
Just for a sec. I'll just. Oh. Let's see.
Where are they on the dive?
That's
T4.
maybe we're going to look
Third.
because we'd like to arrive to a point where it's a little bit flatter. It would be easier for us to
The slope?
slurp, to maybe try the dive one last time, or yeah,
How much dye is left?
it's impossible to know. Or
Yeah. I think you can see the
yeah,
Okay.
yeah. So I think there's enough left for
One.
a last try.
Yeah.
But how long on the transect are we there? I can't judge it from this application. Okay. Oh, good.
Yeah.
Yeah.
So it's 500 meters.
Mhm.
So it's done 150.
Okay. Thank you.
Thank you very much. Depth now?
This is the
633.
This is the profile. So we're I think we're here now in this stretch from 6:30 up to 6:10, and then it will
Then it
again.
levels off a little bit at 6:10. Okay.
So it just takes a bit time.
Yeah, going down.
There's a very short period where we go a bit down, and then we
But not when I'm working.
and then we're increasing. We're climbing again.
Are you sure? Okay.
Do you need anything?
No, good.
I'm going.
Just turn to port now because we're going downhill.
Okay.
So now we're got something.
Look at the nice anemones.
Is it possible to also to stop in on a downhill if it's not too steep?
No, stopping is not the problem.
Okay, it's good to know.
Back the scene first.
Soll ich wieder reingehen? Ansonsten würde ich oben ein Stündchen was gucken.
Look at it. It's looking at
Mm.
it. Mesmerized.
Yeah, I'm going to try and put some full thrust onto it.
It's going to be huge now.
Oh, there's another one. So again, the distance between the lasers is 10 centimeters, which means this fish is maybe 20 or 15.
Yeah. Look at the sea. Is it a sea star?
This?
Pointing up. No?
Or a sponge.
That's what I thought. Or a sponge, exactly. Can't see.
Oh, sorry. Wanted to.
Are we stopping for something specific?
No. Just looking.
Do you mind to zoom in a bit closer? On the lower part here? Yeah, and then
Here?
Yeah, and then go full zoom.
Yeah, that's the next one. So I think these hairy sponges that you see, brown, here and there, this is probably the genus Tenea. And it's probably Tenea valdiviae. And that species has roots to settle itself in soft bottom, usually sand. But here it settles pretty well in the spigomat as well. So we haven't seen that species very much since the beginning of the expedition because it is camouflaged in brown, and for me to identify it I have to see the openings that are, and I can tell they're not as globular as Geodia. So Tenea. And some lollipop sponges.
Can we stop this movement, please?
There's a little snail.
There's several.
Thank you.
One is also here.
Oh yeah.
Yeah. One we just left outside of the night. Yeah, there were quite a few.
Okay. Oh yeah.
See?
You're right.
Yeah. I locked them.
3 snails on this screen right now.
And I'm sure many more small ones that we can't see. Shall we continue with that?
Yes. We're ready to
Move on.
move on.
I won't log it. I think it's the same.
Yeah, possibly. Yeah.
Is this your second time in the Arctic? Or on an benthic expedition?
I would say second time, yes, for Arctic. Yeah. Yeah. And you?
Yeah, I've been to Greenland before, but was on a very small vessel very close to shore, so I had not been into the
In the deep
here in
sea?
the high Arctic deep sea, not with an RV. Well, I've looked around Iceland quite a lot, but it's not quite Arctic.
No.
So, the North Atlantic, and the fauna is quite different.
It's a bit challenging because it's really not easy terrain to stop yet.
It's so covered with life. I'm still amazed by this.
Come on.
It's so covered with life, with living organisms.
Yeah.
That are so many macroscopic organisms. Seeing them here.
Yeah, maybe at some point it would be worth scooping a piece of the spicule mat. Is it a squid or? Yeah, it's a squid.
I'll put down zoom for that.
I can zoom out. Not easy to follow. No. We've seen lots of squids.
Today, yes.
Yeah. Yeah, on this image you can see the extent of these sponge grounds. They cover really large areas.
Well, it's the shallowest sea mount for quite some distance, so
yeah, it's a different water mass and different depth.
How would you like to carry on with this? We need to go straight down there and
Is it a bit flatter on the?
it looks this stuff.
It looks a bit flatter. Yeah. Maybe we can try that. Sure.
Two more sea stars. Sponges.
Wow. Could we get close to this?
To this left patch here.
do you want to face it?
Yeah, exactly.
That would be great.
Yeah.
Dice come out.
Oh yeah.
Oh, where's that come from?
It's from our.
Okay, no, I know, of course I know where it's from the tank, but I'm surprised that it suddenly gave a little cloud.
Unless the hoses.
I'd still love to try it after your sponge experiments to try it on another
Yes, come from
animal.
Sure. Sure.
Yeah. Just sort of not like.
The fish are on the seafloor. Both of them.
There's more squid. Oh wow.
Right, let's wait. Let's wait
Yeah.
until he's stopped and then resume. Turn on the laser. It looks nice.
Mhm.
Tell me what you think.
Yeah, could you turn on the laser? Oh, it is turned on.
Oh.
It is. Yeah, it's like
Oh.
it's there.
Ooh,
Just very
yeah,
far.
Okay.
Look at that,
That is a lovely
wall.
wall. Shoo.
Wow. So I guess with a fisheye camera, this must be beautiful as well. Look at that. Do you think we can
Have you stopped?
settle or
You stopped?
Floating.
you're
Okay,
floating?
you're floating. Fine. Then we'll zoom.
Yeah, there's perfect condition for it.
Perfect.
If you want me to sit
No,
down,
no,
I'll
it's
sit down.
fine.
You can,
It's fine. It's all good. Yeah.
So there's all the key players of the sponge ground here. The tube glass sponges.
Turn the laser off, please.
Could you turn off the lasers, please?
Oh, there's anemones. So many.
Thank you.
Oh.
Yeah, we're leaving it. Is it possible to stay a little bit down? Or
I
down?
can, I'm 3 meters up, 4 meters up, so if I sit down,
Yeah.
I'll sit down and see what
Thank you very much.
But
We'll
actually,
just
if I
try.
come back, you can zoom in a little bit better than that.
Yeah.
That would be great.
Yeah.
There's more of this yellow one that we had yesterday as well.
We are at 636 meter depth.
Yep,
Thanks a lot.
that's right, it's too far.
Hmm?
Too far away from the wall.
That's okay, look.
Too busy watching. Water flow.
Here it's perfect.
Yeah,
Yeah.
Thanks a lot.
it's all yours.
Thank you.
Wow. Super beautiful. Let's enjoy that view. There's a squid, a little squid on the left, a grenadier fish in the center.
Read Jarvie. Can we stop the ship, please?
What's that fish doing?
Huh? Did you want to break?
And is that our first urchin here?
Oh yeah, there's a sea urchin on the bottom.
Wait.
There's a logging. And And there's a sea star on the wall. Okay.
Yeah.
I'll zoom in a little bit more, yeah? There's quite a few ascidians. What are these? Little red things on the floor.
Are these your animals?
They don't move. Maybe it's a decapod resting. That's what I think.
Yes, they are. Yeah, that's.
Yeah, the decapods to the left. No, but this is very pretty. We're looking right inside. Oh, hello. Hello. Hi, gorgeous. Do you mind if I tilt it up a little bit? May I?
Yeah, absolutely.
Look, there's another one.
Oh, yeah. There's the sea urchin.
We haven't had many.
Nope. What is this?
I wonder as well. I wonder. And also, this hanging down, what was this? Another anemone?
Oh, yeah.
Or
Yeah.
we're wondering what left of the sea urchin is, like this white little lines. Now look at these pink things. That's another anemone.
So the substrate here is essentially sponge spicules, sponge skeletons. So when the sponges die, the remains stay, and it's a lot of glass needles that accumulates for years and years and years, and then it becomes a substrate for many other animals. Maybe we're in the territory of this fish.
We'll leave again soon, don't you worry. Actually.
Wow,
I want a full,
this is
full anemone view here.
amazing.
It's like
How beautiful. Doesn't it make you love sponges?
It certainly does when you see it in real life. Yes.
This competes with any tropical reef that you've seen.
Oh, in German they're called Blumenttiere, so like flower animals. Not the sponges, no. These other beautiful pink anemones. So
Yeah.
you understand why. So I'm just playing very little. I'd like to look at the sea star a little bit closer.
Yeah.
Yeah. I like how these fish come and visit us. All the soft corals. There's so many different sponges.
Mm-hmm.
Would you like to slurp anything?
Not at this moment. I wonder if we could try one last dye experiment on one of the sponges that's really close to us.
We certainly can, yeah.
So that you were looking for some big whales.
Yeah, but we could try another completely different species too. I think of these ones. They're geodias, and they're
This one?
yeah, or the one below even. It's two sponges on top of each other, I think.
This one? I'm zooming in.
No, they're really close to us.
Okay, you
Yeah.
do the centering.
So I think that one. This one. Ah, okay. It's maybe a bit too complicated.
I'm not sure. Maybe it's even easier when it's so close.
Here?
I don't know. It's up to you. But it's
Let's
nicer
see.
to see, and then we can have the
Yeah.
Assyrian at the same time.
Yeah.
And you have the other sponge. I think that's a perfect one.
Okay. Okay.
Which one?
Just
so,
weird.
yeah.
The white one.
No, the whole thing. So if you could put it in that little hole. Yeah, exactly. Yeah.
Okay.
We're gonna try one more dye experiment. This is fluorescein, and this dye should help us to see if the sponges are pumping.
Can you tip it a little bit further so that it's centered? Yeah, no, then we get the sex name. Okay. No.
This is very delicate to do this experiment because you want to be right at the base of the sponge. So here we have a lot of stuff growing on that sponge. Soft corals, other sponges in yellow. Glass sponges are all the tube, white sponges, and they're all growing on top of this large sponge.
Ledge. I need to go behind this one, so. In front of me, just up the way. They're back, is it?
Yeah. Let's see. I mean, we could try there as well. Yes.
Where were you thinking of?
I was thinking more of that
Down
little
there.
hole, but I guess maybe if the current is coming
That's
like
what I'm
this,
thinking.
yeah, then I think your spot is better. Yeah.
Mm.
Yeah.
It's up there.
Yeah, let's try it like that.
I think it's still there, and so. So, squeeze it, yeah. That's fine, that's all. So.
So this is really a trial on the geodia. Geodia are much more massive than glass sponges. We'll see if they pump as much.
Well, it's a geodia itself, but there's also lots of critters sitting on it.
Yeah, all of the
So-
filter feeders will be able to.
So we'll see what
Pick
they
up.
do. The other sponges and the soft coral and the Ascidian.
Push the shut.
Yeah, you're doing well. It's coming.
So we're pushing on the bag, on the pouch, to make the dye arrive at the tip. This is a fluoroethane, and it's used to track movement of liquids in humans, but you can also use it in ecology, like here.
Is it actually being sucked in here? Look at it. Look at the side of it.
Yeah, I can zoom in a bit.
And to the right. So thank you. Yes, look, it's coming out here.
Yeah, it seems to be coming out from some of
We'll just observe it.
Yeah, it's coming out
Awesome.
from some of the glass sponges,
Yeah.
the little ones in front. Yeah.
And to the Ascidian on the right. Look at that one.
Yes,
I think
it's being taken up by the Ascidians as well.
Yeah, how cool is that?
On the
It looks
left.
like a little smoke. And the bottom one. That's a sponge. It's an Ascidian. Cool.
Yeah, it's taken up really well by this one.
Looks
This
like a
one
little
is-
smoker.
Yeah,
A little green smoker.
as for the geodia, it's not as visible.
Maybe it's just slower. I'm trying to remove this. Thanks.
I know it's
Yeah, I'm not sure yet.
it's difficult.
Yeah, but look, isn't
Yeah.
it sucked in? It's hard to see because we don't
Yeah.
know how much distance there is between.
Yeah.
It's very interesting how it goes to the sea floor on the right side as well. It's a pattern.
Oh, this one is a real smoker. The
Yeah.
little one. It's really pumping.
Yeah.
I wonder if this is coming out from the geodia. Because otherwise I don't see where they would come from, because the syringe is not there anymore, but it seems that it's green on the geodia. So I think it's pumping out now. The geodia, you can see it's a little bit green inside on the top surface. I think it's definitely, because it has several holes. It doesn't have one big hole like the glass sponges. So this is what we should expect to see, green coming out from different parts of the geodia on this top surface. Yeah. So this is very nice to see as well. And it took a little bit more time for the geodia to process the dye. Still more coming out from geodia.
Yeah, and also still some of the dye on the sea floor or close to the sea floor.
Yeah.
Which is slowly coming up.
So that's confirmation that all of these sponges are not sitting on a rock. They're really sitting on the living animal. It's a sponge.
Setting.
This,
I'm really pleased it worked.
Yeah.
Really, that's great. And look at this obsidian. Look at that. It's full.
Yeah.
The little one has stopped.
Yes. It doesn't have any more dye probably, but geodia keeps on going. It has taken up a lot, apparently.
Would you mind if we now zoom in, for example, on that part? Or on this one, just to see? No.
So there's a whole little ecosystem growing on this geodia. Other sponges, but also anemones. This is probably a calcarean sponge, maybe. what's the name for that?
Caprolide.
Caprolide.
It goes to Trump. So it's an amphipod,
Yeah.
like centered.
I don't know if you can spot the little crustaceans moving in the anemone, almost in front. I'm going to try to center it.
Well, they're two above each other, so you can center that or you can center that one.
Oh, I haven't seen the other one. Yeah.
Well, the top one is probably the female because it's got this convex shape of the middle part. That implies the eggs. It's really hard to see.
So they look like little white
if it is.
twigs that are folded. But these are small crustaceans. What is that? A hydrozoa
I
growing
think
on soil?
on the anemone, I think, yes.
Yeah. Wow. And the geolia keeps releasing the dye. There's still breed patches here and there. Is there any other place?
When we zoom out and then go a little bit to the left and zoom in again.
So this part.
Nej. This part, we've been up here, haven't we?
Yeah.
Can we zoom into that part on that?
Oh, what is that? Isopod? Or it has very long antennas.
I'm not sure. It's definitely a paracharid crustacean, but I'm not sure if it's an isopod or an amphipod. Are we looking more sideways?
No,
And
I
feel
think we're looking really at the top of it, because you see the two antennas are like this.
yeah, but maybe they've got four antennae, and two are bent down. I'm not sure. I really don't know. Maybe it is.
Oops, sorry. Just going to.
I'll log it as an isopod. See the other caprolid on the left here? Oh, that's the same one.
Yeah.
That's the same one, but you can just...
Yeah.
Okay.
And I'm going to show you another tenea here. We have a very nice tenea, so they're usually rooted in the sand. So this we have only collected only one, I think, from the beginning, and it was a bycatch. So, do you want to try to slurp something here? Because if you are interested in slurping, I can slurp one or two teneas.
Oh, I'd be interested in the caprolid, but that's a tricky one to... It's super small.
Have you already slurped capillite? They
No, we picked them from the sponge, and I'm not sure how they, because that's not how, they don't sit inside sponges like this. A typical sit in the outside in the current. And you have this wave-like movements, so I'm not sure if it's going into one slope, if it would. So we can try if you want to, because we're sitting.
Yeah, we can try.
Slope, yes.
So I think we could try to slurp. There's two little crustaceans sitting at the top of this geodia.
Just go down and see if the arm can reach up to it.
Yeah. Maybe I'll zoom out a bit as well.
So I have to swing around and touch.
That's maybe a bit far.
Okay, 1 second, I'm going to suppose my T4, you know. You're finished with the dye?
Yeah,
Yes,
finish with the dye.
thanks a lot.
Take this out of the way, so.
Yeah.
Have a look at sister at the beginning. Maybe not. Sister.
This is Sergi. This could be Sergi's complicata. Oops.
Oh, then it's flipping business here.
Yeah, I think.
They can all go in the same slope.
Yeah, everything can go in the same slurp. No problem. What is that?
Oop.
We're moving.
Just a little bit.
To get closer. Okay. Yes. We're preparing the ROV to do a slurp. A slurp is a little bit like a vacuum.
Like a vacuum cleaner.
And it's ideal to catch something without damaging it too much. It will end up in a compartment at the end of the ROV. We have 5 different compartments.
Go with the tube. Do you want to put the colors out?
It's a second one.
Yes.
just double checking.
Yeah, it's inside. Yeah. Yeah, I think We put it back when we started doing the dye experiment. Another option could be to. No, we don't have the. No, we don't have the oven.
Just have to push the gate and the nozzle around.
Should we write cost one?
No, definitely. We'll do all the records. But this is, what is this here? Nothing in it.
No, we didn't write it.
Oh.
But we did fill the other one, so we can copy. You know the other one that we do always at the beginning.
Yes, I know which one.
So we can copy later on.
So class one. This is slope one.
Yep.
So, and the depth here is 640 meters.
Yeah.
And here it's like the time, which is 2042.
Hey, little zoom there, back up.
Yeah.
Zooming in?
Yeah.
You want this?
Yeah. So what am I looking at here?
So there's two crustaceans, one here and one here, really small.
Thank you.
You won't be able to see them going up there.
Yeah.
Too small.
There you go.
Yeah, good chance that you've got them.
I think.
Oh. Yeah, good chance you got them. All these sponges will regrow.
Yes.
The sponges, these amazing animals, will
They will be
Go back.
able to regenerate and heal. And we got some sponge samples as well. You can write.
like an apple tree. I would like to. Do you want the one for Sergi? A little bit.
Yeah, but
Or the top one here.
Yeah.
Hmm?
Yeah.
That was great. We can keep slope one and we'll sample a little bit of a sponge for
I'm trying to see where it was. I think it was.
that one. I think it was more to the right.
More to the right.
Yeah, no, let us just zoom out a little bit so we find it quicker.
Ah, no, it's here. That was the big one we saw.
That, you want
But-
that one? Okay. Is that an option as from angle? It's to the lower left.
Is this one possible?
Let's see. Okay, point on the camera point. That white one.
Yeah, the bushy.
Yeah, if you come left with your camera and zoom in.
Sure. So we're going to try to collect this sponge called Lysodendrix complicata, on which we are planning a population genetic study and a environmental DNA study as well. So we've been collecting several individuals on every seamount, and with genetics we're going to try to see if these individuals are genetically connected. Yeah, it's perfect.
Okay, chamber one.
Chamber
Yes,
one.
yes,
Yeah.
slope one. Exactly.
Perfect.
Okay.
Absolutely,
Perfect.
absolutely perfect.
Turn off.
Could we take that one as well? That's a Tenea.
That's how it started. This whole
The
thing.
clear one, is it?
No.
This one.
No, the potato-looking one.
Yeah, with this little hole. Yeah.
Yes.
Okay.
Yeah.
Yeah.
Oh, it's still attached, I think.
Okay.
Yeah.
Take it down a little.
Yeah. Yeah.
Yeah.
Now you got it. Perfect.
Okay. Is that it for here?
Yes.
Yeah.
Thank you.
No?
Sometimes I wrote sponges.
Perfect.
In the cities. Thank you very much. I think we should fly on from this side.
Okay.
Oops. Which one I want?
Still have some of that.
Okay. So with Rob, we decided to go on the right side here.
Yeah. More to the right.
Yeah. Because the other side seemed a bit too steep and high.
Okay.
It happened. I have it capped in. If I press is it 6? It's like an upper shift. No?
Oh.
How do I unlock it up
there.
there? And then,
Now it works.
okay.
So we're following this wall of sponges and anemones. Can we put the sextant out?
Yes.
Thank you.
Traces look like little landslides. Like the rock
I was
has
thinking
been-
the same thing, yeah.
Oh, did you have the ship on?
030.
Range RV.
What was that?
Yeah, we get a vessel there bearing 030.
No, it's moving out.
Oh,
It was something.
I didn't see it.
But they're quite long, so it just indicates how steep it is if
Yeah.
something rolls
Yeah.
down so far.
Thank you.
Take these odd shapes. many ascidians too.
Yeah. This is flatter. It's a little bit flatter.
But it's Ascidian territory here.
Yeah, the anemones have disappeared.
Tilt down a little bit back up.
Sure.
Just a little bit down.
Like this?
Yeah, see where your glare at the top of the screen is going?
Uh-huh. Here a lot of Asconema foliatum, the glass sponge, looks like a cauliflower, and a lot of ascidians. These brown potato-looking
But
animals.
more translucent. So potatoes already used for your for your sponges.
That's true.
The see-through potatoes. They have a white line.
So we should be here. It's a little bit flatter, but then it's going to become steep, quite steep again, and then flatter here. So the end actually will be quite nice, I think, quite... maybe better to collect things near the end.
But I was quite happy that it was very efficient. Stone?
I think so too, yeah.
You can just fly up there, so...
Paddy?
Yeah.
What is it? Licorice?
Yes.
Yeah. They will be in there. Thank you.
You're welcome.
Are they all for us?
They're especially actually for the lady to the vegetarian. For sure.
I'll leave two there for you for later.
Thank you very much.
Thank you, bud.
That'll keep us awake. Well, me anyway.
speed for just looking so. Yeah, that's not. Oh, sorry.
Hey.
Just wanted to check where we are on that translate. Oh wow, it's still
It's absolutely
Yeah, because
stunning. We're the dye experiment.
It's now kind of okay with the dye, right? Or do you want to
Yeah. No.
Yeah.
No. We're done with the dye.
Yeah, I would say so.
We had one slurping stop, but we have 4 more.
Wie lange das Translate ist? Hast du das Buch gerade mal da?
Yep.
Because it would be nice to cover that translate before we come up. Finish it.
Oh, look at that.
500 meters. And we have, how much have we covered? I still owe you a new load of Haribos, I think.
Sorry?
I still wanted to bring you a new
This have to wait till after
This is all
midday
in.
tomorrow.
No, give it to him. This is all
Timing it well.
strategic.
Yeah,
You can give it to him. Although we have to keep them awake and happy. It's like small children.
Yeah. But I heard Andrea likes the vegetarian.
Yeah, I know. Do you think?
Should I open them now or what?
Yeah, absolutely. Paddy is here. Then we'll.
Kind of gone now.
Well, we're not sampling at the moment.
Or are we live?
Diese sind vegetarisch, die vorderen.
Dashboard down.
Advertising on
Yep.
We're sponsored by Haribo.
Can you spell it? What was the name of the brand? No, this is a
Sponsored
sponge.
by
Harry's sponsor.
Sponge bias.
That's house bundles.
Yeah, the sour are the best ones.
Shall we swap? Just take
Too
this.
sure?
It's just for
Sure.
draft reasons more than
Anyway, I
anything.
think Jenny will come soon.
Just thank you. There's no further meaning except for getting the draft in a different
Yeah,
spot.
yeah, I totally
This-
get it.
Hmm?
Yes. I'm glad we decided for that transit.
Can you see the lasers?
No, I don't think the lasers are on.
That's my new skill.
Our DNA.
Yes.
Oh.
That's quite funny.
16S?
No, it's a rough DNA. It's my RDNA.
RDNA.
Contaminated. Paddy says so.
He's one of us now.
Oh, careful.
raffle. The person who wins the raffle gets to fly.
I think that's the night shift. I think that's the night shift.
So,
Are we still live streaming?
Yes.
Okay.
This is real life.
this is
Sea urchin.
With very few sea urchins.
Yeah.
With very few echinoderms in general. We had the girdle stars. They came en masse, but in patches.
Yeah.
And now we are back to sponges. And sponges and more sponges. Another sea star coming up.
Marco, have we ever sampled that sulfur yellow sponge? Is there, do you see any possibility? I think it was in Schulz Bank, right?
Yes.
Yeah,
Yeah,
it is?
yesterday morning.
But that was another one. There's two yellow ones. There's Aplyzilla, which prefers rocks. It's not here. And the other one we have here is Hexadella.
I think that's the one I
Yeah. We haven't specifically sampled it. Oof, it's getting dense. Look at that.
It's very, very nice.
We
I
are
love how you can't see any sediment.
at
All grown
612 meter
And now-
depth.
And more the soft corals are back. Can you?
Mm-hmm.
So. I can show you on the profile. Do you remember the profile going up? Having
Yeah.
this little bit? Then that was acidial dominance. Now it's going up again. Now it's back to sponges.
was going to say it again. But then I think the style
Actinophore.
No, it's actinophore. You see it. You see how this, it's got these shimmering lines.
Yeah.
Rippenqualle. Also, I think it's super nice.
Is that the last until 2 morning?
Ja, das ist auch schön.
And I think that's when we should cap it. Yeah. To allow for one more dive with full sampling equipment. That we then need to finish around midday, roughly. Yeah.
All good.
Your call, guys.
Yeah.
Of course, we're slower when we stop here and zoom in, but that's of course an interesting part. So I don't know what the priority is, to finish the transect or to
Ready to
finish
start line one?
Well, of course, to also to go sampling and look closer if you feel like there's something new to
As always.
see.
Well,
You don't have to rush the transect. I'm just
Thank you.
Okay.
wherever you are at 2 o'clock. That's where you just go.
That's a bizarre one as well, isn't it? It's white.
That's Asconema. Yeah.
But isn't it a bizarre shape?
Yeah. Yeah.
It's a little hedgehog.
See ya.
Just want to zoom it just so the other cameras and hoses are out of the way.
It's
going down.
Sister.
It's still there. I don't have anything in my book.
There's another squid. Or is it? No.
You should love it.
Good. I'm glad I brought them.
Did you taste them?
Enjoy. No, I don't want those. I would like the other one, please. Thanks.
Are there? Could we have the lasers on? I think they.
No, they are.
They are on.
They're on.
Okay. It's so pink. Maybe I don't.
We are going down.
Down slope slightly, Marco.
Ah. Is that fun for us? No? Oh,
Super
yeah.
pretty. I was so focused on the other one.
Yeah. Me too.
Maybe just a sponge structure next to each other. Yeah, it is. An odd angle.
Oh, that was how did you call it? Angel.
Ihres Engel. Like it's a mollusk. But I don't know the English name. Either was fairy or an angel.
I think it was angel.
In German, yeah, Meerengel. So I really don't know.
No, I'll just write mollusk swimming.
The flying mollusk. Well, then you think of a bivalve.
Yeah, but I wrote angel, so
yes, then it's fine. Can I offer you something? Any supply?
Hmm?
Do you want anything to eat or drink? Or
No.
Or swap? Miss Jenny? She'll do databasing unless we tell her it's entering data.
Then I'm happy to take a small break. Yeah.
Oh, I don't know.
So we are currently going down a bit. That's why the camera is more difficult maybe to position in order to see the animals.
You guys just tilt down a little more?
No.
Later down?
Mm.
Back up a bit. That's good.
Yeah, I'm just trying to get this section out of the way when
Yeah.
flying. That's why I'm
No problem.
higher than usual.
We'll call down.
Now the occasional anemone is back
Mm-hmm.
and some fish. The sun reflecting the wind.
Yes.
Unbelievable. It's been really well. It has not been sunny very often, and apparently we're in the container now, and it's so sunny outside that there's a reflection on the winch and on the metal cable. It results in a very bright screen. So it looks like a computer error. Because we're so high in the Arctic, there's no sunset, so it's always light. It's very nice.
Yeah. So we can see the midnight sun, which means we can see the sun at midnight. It doesn't set. It stays and then goes back up in the sky. And that is because we are beyond the Arctic Circle.
That's a light crop.
Tell you what,
Do you want to go droning?
So sure.
the sun is out.
And yeah, we'll crave some. Yeah,
Yeah.
sure. Well,
Yeah.
have a nice break outside.
You could open the door and then see the sun. Yeah, it's brilliant. Very bright.
What's the temperature? Yeah.
The temperature is currently 0.1, so it's positive.
Okay.
Positive temperatures here at 607 meter depth.
Yeah. That's our challenge, getting these samples back to the surface, is keeping them cold. So these, you're laughing. Often people think that it's a pressure difference that.
No, I'm laughing because it's cold here.
Yeah, it's super cold. It's super cold here, but this warm surface water of a few degrees is challenging for these animals living in 0 degrees water.
Yeah.
So that's why we, before the ROV comes on board, we pre-cool water to get it to 0 degrees, so the animals can stay alive
Mm-hmm.
and then also be photographed. So keeping their colors and all legs and appendages looking as beautiful beautiful as they are. Because we've got the photographer Solhan Sankal on board. He's a deep water photographer. And does an amazing job. So a strict rule is no air bubbles on the sponge. So keeping them
them
in the water
at all times it's a bit challenging sometimes.
Yes. But possible.
But possible.
Yes.
We've done it.
Yeah. Is that another 600 to start?
Yeah.
Many hours? How much longer do we go downhill?
Oh, it's
coming out
coming
now.
up a little bit.
Okay.
No, we're going up now.
Okay.
6.5.
It's that line there.
150.
Oh, yeah.
150 till end of transit.
Yeah.
You can better stop.
150 meters till the end of the transect.
Yeah, because we're zooming along now.
I mean, we can't stop and look at things. I mean, it's fairly flat.
Can we do that?
Yeah.
As well? Can we just stop here and thank
Look
you.
at the scenery. We're gonna have a little break to enjoy the little animals of the sponge ground on the top of Schulz Bank seamount at 603 meters. So to settle, we'll create a bit of dust clouds, but we're gonna wait for them to clear out.
And we have not seen anything in particular. Just want to have a closer look.
Yeah.
And as soon as it settles and we've seen the laser, I'll turn them off. But it does look very nice. Yes. So is this why there's still this coming? I don't know. Yeah. Okay, I'll just move back again.
Oh, is that an anemone? Is this one of the really long ones?
That has just pulled itself together. Okay, should we zoom?
Yeah.
Should we wait a little bit more? So can I turn these off, please?
Yeah, these
Yeah. Thank you.
There.
One of the children in a school interview that you gave asked about making glass. I thought it was a really interesting question.
Yeah.
How they can produce glass in these cold conditions.
Yeah. Sponges extract silica from the water column to make their skeleton of glass because glass is you need what is it called? Silicium. I think it's silicium. Yeah, and
sure, it's the same
oxygen.
Same as our windows.
Exactly. And then they manage with a series of enzymes to produce their skeleton. While humans, to make their glass, we need sand that we melt, and we need 1,500 degrees, so a lot of energy to produce our glass. So there's a lot of research going on to understand how sponges do it at 0 degrees or at fairly low temperatures with the help of many enzymes, so proteins, to accelerate the process.
There's lots of interesting research going on for sponges and how to, I don't know, make use of their characters. What's this?
Yeah, looks like a nudibranch.
Jennifer! Jenny, where are you?
Did you?
Yep, I did
right
let her
to-
know, but she ignores me. Well,
Yeah, and that's hidden here is Geodia parva, but it's so it must be massive hidden. But for once we see
you
its
talk about
oscules.
this dotted unit next to the, because the viewers can't see the pointing.
Yes, sorry. On the left hand side, there you can see several little holes and that's Geodia parva. So it's one of the key players on this sponge ground. But it's camouflaged because it's brown and it's in the middle of everything.
So a nudibranch is also a mollusk. It's like a gastropod, like a whelk, but with no shell.
Mm-hmm.
In that group of animals, and we haven't seen any so far.
We caught one on the first.
Did you?
Yeah.
Oh.
Yeah. We have one.
Oh,
I think it's the same species.
okay.
Yeah, it looks
Not
very
something. Okay, I wasn't that one.
Yeah.
Here you see those
And-
whelks. Look at them how they're.
beautiful. They look like a different species than what
They
we've
do.
seen. Yeah.
They do. We had these ones are more striped,
Yeah.
and you can see the siphon poked out. You know the lower snail in the picture. That has this unit, this cylindrical picking out. Oh, the tape change in 10 minutes.
Yeah. Tape change in 10 minutes.
And can sense smell of that. And look, there's a little crustacean coming behind. No, there she is.
Oh, you
Now we left.
you have to
We left this
sure.
beauty in the picture.
Yeah.
Excuse me?
Yeah, excuse me, we left this here, so now we can move on.
Oh,
Yeah, just for you.
So, oh, these are different species.
Yeah.
That's exactly
We
what
were
we
just
just said.
discussing about
Yeah.
it.
Can
Yeah,
we slap it?
Yeah,
I mean, we're flat and settled, and
yeah. Let's have a further look and then decide. Okay?
Paco, we can swap. You've been here for a long time.
like this, I can
Yeah.
eat something.
Yeah.
Enjoy.
So we have slurp one, which is quite full, but the rest is empty.
Okay. This is very interesting.
Isn't it? What are you eating?
And
You see the sea star? Can you please log nudibranch and sea star?
can we go back to the nudibranch?
Yeah, not yet. I'll find it. Now look at this, all these things. Yeah.
Wow.
Putting out.
Look at the sea star and the sponge.
Just having its arms hanging over the side. Of course, for our pilots, it's more relaxing when once the RV sits down and we're not yet sampling. They can have a little break too.
Do you know where the nudibranch is?
No. I don't know how many more you might find.
They're all hiding.
I knew you were coming. I think they're all camouflage now. Look at this. I can't zoom further.
That's all right.
What's that? It's a dark reddish one in the middle.
Tja.
I don't know.
So pretty. So where was the nudibranch again?
I don't know.
You know exactly where it is. Are you joking? We need to find these snails again. They're
I think the nudibranch is just to the left. Look, it's just outside of the screen now. I think it is. Nope.
it when I came in.
No, we just coincidentally discovered it. This is not the. These are not the whelks. I know. You meant this part. Let's look in the sponge here.
What's that?
Very okay, but I wanted to look.
This is the soft coral.
Isn't that nice? Well,
Well, we need to find the two whelks again, and then we find the nudibranch. The thing is, I was thinking that you had looked at it for such a long
Not
time
at
that you
all.
got tired of it.
no.
Okay,
Just started.
go in that direction, please. Yeah, that there. There's the sea star. Just wait a second. Can you tilt a bit down? Tilt a bit up. Sorry. This sea star was in the picture when you showed me the nudibranch. Sea star and two snails. Are we moving?
I'm wondering.
No?
It seems that it feels a little bit like
It
it.
feels like we are moving.
No, we're still here.
It's a ship, maybe. But it's a bit
Because we're turning, aren't we?
Okay.
Bridge
Let's
over
go.
without stop.
Let's go again.
Thank you.
Well, just the whelks that we saw were definitely a different species from the ones we have had so far.
I agree.
I would like to find them again.
I have to zoom out a little bit to
Yeah, it's so dense.
and it's also difficult to get the size. You know, I'm not sure if I'm looking for the right size spectra. Like that.
Oh,
seen that?
no. Can you zoom in on it?
I'll try.
Wow. I didn't notice. I was now very focused on the small things.
Yeah. So I'm going a bit further because I want to see the sponge underneath what's sitting on it as well.
What's that?
What are you asking? The starfish, the soft coral, the acidian?
This.
One? Yeah, that was what I was trying to see, but I can't.
No.
No, I don't know. It's so dense. So we might take a little while looking for these two snails.
Okay. Just,
No.
just,
It was just in the beginning, and I would normally zoom in from an overview. picture without the sextant inland in it.
Yeah, but the sea star was one of the first things I saw. There it is. There's one. There's one. Do you see it?
Yes, but it might be a different one.
Yeah, it's not the same spot, but
No, it's not the same spot. This also looks very different to the ones that we had so far. Huh?
I think we should sample it.
Okay.
It's a different species.
Yeah, I see. Okay,
We're trying.
We'll try. Can we try slurp 2, please?
Yeah.
And having...
It's getting tiny now.
I can't really see it on...
Zoom out and then I'll have a look on here.
I can see it. I can see it.
Let's swap and you...
Yeah.
And you direct the...
It is...
It's in front of the camera. Just have a look. That's in front of the camera, so that's pointing. That camera's pointing that way, so that's pointing out that way. So
Stop.
yeah. So that is that.
Yeah. So it's here.
Oh right. So if I put that over there,
It's here.
it's very here.
There.
We'll zoom in a bit and I'll see if I can.
It's so small. I think it's that.
That little thing,
Yeah.
that little ting.
Yeah, it's
Because every
tiny.
little ting.
Just turn the lasers on, just for size, just a second, when we zoom in.
It's actually no. It's actually that little thing. Exactly that one.
Okay.
Yeah. So we have an Assyrian here. We have an Assyrian here,
All in one go.
and then you have. I only want this little snail. Maybe it's not possible.
Oh, you'll get it all.
Yeah.
You will get it all.
Yeah.
Well, shall we do the tape?
Oh, look at that.
Yes, see that. Shall we do the tape
Okay,
change
now.
first?
Yeah, we can.
Because it's in 5 minutes.
Yeah. Yeah, we take
So,
that one.
take that instead.
Yes.
Okay.
Is that better?
It's easier.
Yeah. Okay.
We do the tape now, will we?
Sorry?
Do the tape now?
Yeah.
Okay, stopping in 3, 2, 1, stop.
I just wanna see.
Quick, it's running away.
This will be nice for our photographer too.
Yeah, I know. You let me know when it's...
Yeah, one second now.
Yeah, no rush.
Okay, start recording in 3, 2, 1, go.
No. Yeah.
Can we call the trailer, please?
Sorry?
TriLab ROV, that's the Keg Pro changed and started recording again.
Okay. You still got it?
Yes. Yeah.
Yeah. There we go. Perfect.
So Paddy, should you choose to?
Can you zoom out on yours and just follow me up?
I can.
Now I'm going to bring that sextant camera in, guys.
So where am I going?
Move the arm out the line.
Spider lasers, Jenny.
A bit. It's on here.
Okay, that's whatever they are.
Okay. Nowhere near it.
Yeah,
No.
looks very good.
So you're going to have, ready? Go.
Okay.
Very good.
We have
Yeah,
the royals.
Yeah.
Zoom too.
Wow, thank you.
Okay.
That was perfect.
Would you like anything else?
Yes, but we're looking for it.
Yeah,
Okay.
we're still
I have moved
searching.
the arm out of your way.
Thank you.
You're welcome.
It's so dense here, and it's also pinkish in color, very similar to the soft
Feed your
pole.
boots, ladies. Feed your boots. We're here for another few hours.
That's true.
Well, one
So we were definitely more
Yeah,
headed
just
to.
zoom in.
Yeah, but the nudie ring is there. Oh, look. That's a solasta.
Temperature.
Sorry, can you go down again?
Yeah.
Clear it.
Can you zoom in a little bit? Just
What are
here.
you looking at?
Can you zoom in just once
Oh yeah,
here?
I was up there earlier. So I saw that 1, 2 and 3 and I saw that.
Look at all the amphipods.
This is remote protection, basically temperature. Must be
No, I'm good. Thanks.
fine.
It would be nice to have 2 snails, one for morphology and they're hard to find. How come you had such a good
Skills, baby. Come on.
skills, baby?
More skillful to refine things, not to.
I think it's okay. We might find another one.
We might not.
We might not. They're really good at hiding. I still can't believe that you showed me this nudie and then just zoomed away.
Because we just started zooming.
I would have loved to see this a bit longer than like 2
You
seconds.
should have come earlier.
Okay, now we're starting. Shall we fly on? Shall we cover some track?
Well, we don't have much. We're 4 hours for 150 meters. So
Oh, we
we're
can
allowed to zoom.
Easy.
Okay. What's this? Is it decapod?
It's not a nudie rank.
No, it's not a new. Can you look? Open your mind for other.
It disappeared.
They are scared of you. Just.
What is going on?
It's running off. Look. So, in slab 2, I've got a snail.
Yeah,
1.
one snail.
Yeah, that's fine.
Yeah. I'm gonna look for a second one.
There was one right at the lower part. Towards
Really?
Yeah, towards the one that you also saw that it was hard to sample.
Where though? No, I lost it again.
I think you have to go a bit further to the right. And now go down as much as you can.
Ah, hello. Yeah, let's go for that one. We are now looking at that snail, which is here. Can you zoom in? Thank you. And that can go in the same slurp with its body, so they're together. We're sampling these snails because they look very different from the ones we sampled before. And we're taking 2 specimens in order to have a replicate, like to have not only 1 specimen, but 2. Because we want to do different types of analysis on these, amongst others DNA extractions, and it's always good to have a replicate.
So there's the laser on it, Jenny?
Yeah, it's basically right in the middle of the 2 lasers.
Can you zoom in?
Yeah.
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 Done it? So, it's in the box.
Okay, yeah.
Is it gone from the ground?
Yes.
Okay.
Thank you.
That's in there, sure.
Yeah.
For me, that
Fine.
would be all. Just slurp here.
That's fine. So we can fly on.
Okay.
Still the arm.
I just
Go ahead.
have a-
Thank you.
Thanks a lot.
Bitte schön. Let's make this happen, Rabo.
Where are we going now?
Continue the line, please. Sorry?
I think we're moving.
So to the right.
That was a very pretty stop.
Very pretty. You choose the next one.
Okay. Do we want to swap again? Do you want to continue zooming?
Yes, I do.
Yeah. Go ahead.
I do.
Yeah, I feel more. I'm more in a logging mood than in a
That's good.
zooming mood.
Well, I'm in a not under the cold airstream mood.
Yeah, I get that.
You straighten up the T4 mistake there.
Hey, you should be distributing.
I had a tea bag in my pocket.
You want to just straighten up your camera, guys? Back to the main.
Onto it.
Yeah. Sehr gut, ja.
Dankeschön.
Dankeschön.
Bitte schön.
Kannst du Deutsch sprechen?
Yeah, probably take the
Ein
camera
bisschen.
there for a second.
Ja.
Need this size.
Da Jenny und ich beide aus Hamburg kommen,
Okay.
haben wir gestern eine
Ja.
Stunde hier im Deutschen unterhalten.
Wir können auch jetzt ein bisschen Deutsch plaudern. Oder hast du nicht so?
Das ist alles recht.
Ja.
Wir zoomen ja immer gerne rein und deswegen haben Jenny und ich die Reputation, überhaupt nicht voranzukommen.
Richtig.
Aber das macht ja nichts, weil wir sind jetzt gegen Ende des Transeks. Und wenn wir reinzoomen, sehen wir natürlich viel mehr als sonst. Can we stop again, please?
If I must.
You may have the pleasure to explore. Und man weiß ja immer nicht genau, was da zu sehen sein wird. Okay.
Ja, gut.
Ich finde es schön, dass wir jetzt nochmal anhalten.
Ich auch.
Dann können wir uns nämlich nochmal das Gleiche angucken
Denkst du?
und wieder neue Schätze entdecken.
Das war die Absicht. Guck mal, jetzt haben wir hier nämlich direkt eine prächtig vor uns liegen.
Oh nee,
Ja doch. So was gedacht.
da können wir ja nochmal reinzoomen.
Sollen wir die in den anderen Slot machen, weil wir sind jetzt, oder in denselben, weil wir sind jetzt 2 Meter gefahren.
Ja, wir können das in den gleichen machen, weil
Also,
es hat sich nichts verändert.
Das glaube ich auch.
Wenn wir nämlich noch ein drittes Exemplar sammeln, dann sind wir wirklich sehr gut bedient.
Ja,
Ja, das machen wir.
das machen wir jetzt gleich. Die finden wir auch wieder. Die merken wir uns
Ja.
jetzt. Rechts.
Wie bitte? Wollen wir das nicht direkt...
Das Risiko gehen wir jetzt ein.
Oh,
Ja, wir sind. Guck mal,
Ja,
ich zoome jetzt nochmal langsam aus. Die
die
sitzt
finden
aber auf
wir.
so einem braunen Fleck.
die finden wir.
Das
sagen wir jetzt.
Das sagen wir jetzt, genau. Aber es war noch nicht so weit drin. Merkt ihr jetzt einen hohen
Ja,
weißen.
oha, das wird gleich.
Das kriegen
Was
wir
ist
hin.
denn da hinten los?
Ja, da hinten. Da hinten? Da Ferne?
Nee, ja, ich war eher so hier. Da.
Ja.
Ja.
Ist zu weit
Anemone,
weg.
oder wie?
Ja, ist aber zu weit weg
Ja,
für hübsch gucken.
na
Wir
gut.
gehen nochmal raus und kommen nochmal ein bisschen näher an unser schönes Gerät. Ich finde das ja prächtig. Jetzt sind 604 Meter.
Mhm.
Wir gehen gleich ganz nach unten.
Okay.
Was ist das hier unten? Das ganz große.
Ich hätte gerne mal so einen Anemonen-Zoom.
Letzte.
Ja,
Das
stimmt.
kriegst du.
Ein Mini-Seestern. Sehr süß. Und da ist noch ein anderer Seestern. Oder ist das ein Schlangenstern auf dem Schwamm?
Ich glaube, es ist ein Seestern.
Ich glaube, es ist ein Schlangenstern. Da.
So dicken Ärmchen.
Ja, da müssen wir